Hey guys, this is Dan Croucher, Field Application Specialist with Photometrics, and today I'm going to give an introduction to super resolution localization microscopy. So why is super resolution microscopy necessary? Why do we need to surpass the resolution limit of conventional light microscopy techniques such as wide field and confocal? Wide field and confocal are great. They allow us to look at things like whole cells and some of the organization within them. They're great techniques because light is non-invasive. It doesn't bother live cells too much, which lets us see real-time dynamics of living organisms. It's also nice because there are so many fluorescent probes that we can easily use multiple fluorescent dyes to see the organization of living cells and even how structures within cells interact. The problem is that the limit of light microscopy is only down to a few hundred nanometers. Many molecules of interest are much smaller, like microtubules that stretch across the cell and are used as transport pathways to move cellular components from one part of the cell to another. Or proteins such as kinesin, a yet smaller protein that sits on microtubules and propels other molecules along them. Something that would be pretty cool to see, but we're limited in our ability to do that. Ideally, we would like to keep the advantages of light microscopy and extend that to sub-100 nanometer resolution, but we can't because we're limited by what is called the diffraction limit of light. This is going to be a quick physics lesson, but it's necessary to understand what we're overcoming with super resolution microscopy. So say we have a fluorophore here, 2 nanometers in diameter. Once it passes through the objective lens, the image we see is considerably larger, some 200 to 300 nanometers in X and Y, and 500 to 800 nanometers in Z. This happens because light travels as a wave, so when it's focused to a small spot with a lens, no matter how good the objective lens is, the focal spot will have a larger size than the actual fluorophore. This happens because the wavefront of the fluorescence emission becomes diffracted at the edges of the objective aperture. This effectively spreads the wavefront out, widening the fluorescence emission into a diffraction pattern which has a central spot larger than the original fluorophore. This was first noticed way back in the 19th century by a guy called Ernst Abbe, who went on to create the famous equation that lets us calculate the size of the diffraction limited spot if we know the values of the wavelength of light and the numerical aperture of the objective. It gives us the theoretical limit of resolution of a light microscope. Here I've put in the value for GFP, which emits at about 510 nanometers, and I've chosen to use a 1.4 NA objective lens, which when multiplied by 2 gives 2.8. That means the theoretical resolution limit of a fluorophore emitting GFP is 182 nanometers. This diffraction limited spot doesn't really look like a flat circle. It's what we call an Airy disk, after George Bedell Airy, who discovered it. You get a bright central spot in the middle surrounded by a diffraction pattern that looks like what happens when you drop a rock into a river. It's this bright central region that gives us our diffraction limited spot. What this image shows is that with a higher NA objective, the Airy disk gets considerably skinnier and therefore increases our resolving accuracy. Essentially, we can see a smaller spot with a higher NA objective, but only to a point, and that point being the diffraction limit. A further problem is that as Airy disks move closer together, they merge with one another. We can't resolve individual Airy disks when the distance between them is less than the radius of the disk. So in 1896, Lord Rayleigh refined the Abbe equation to take into account how far apart two fluorophores need to be to differentiate them to give the new equation. By entering the values again of GFP and a 1.4 NA objective, we can see that GFP molecules need to be 222 nanometers apart to be differentiated. A great example of the weakness of conventional fluorescence techniques is in determining the structure of the nuclear pore complex of Xenopus laevis. So the nuclear pore complex is an 8-protein complex that creates a hole in the nuclear envelope which allows cellular components to pass through. It's 50 nanometers in diameter, which is below the diffraction limit. So if we were to label the 8 proteins of the complex, it would be impossible to visualize them individually. And indeed that's what we see when we try to visualize these complexes using wide-field fluorescence microscopy. No structure can be determined, just a mass of fluorescence. But by breaking the diffraction limit with super-resolution localization microscopy, in this case D-storm, the 8-protein complex becomes visible. Not just visible, but you can even zoom in and count them. So how do we do that? Well, if we can isolate a single fluorophore, and be sure that what we're looking at is a single fluorophore, we can fit that to a Gaussian function where the height and width of the peak are directly related to the intensity on the pixel. That means the more photons you collect, the taller and thinner the peak. We can then use that peak to more accurately pinpoint the center of the distribution. The final localized data point gives a far better approximation of the exact location of the fluorophore, which in some cases is down to single nanometers. In this way, we can break the diffraction limit. The problem is, of course, that just like in the case of the nuclear pore complex, not all fluorophores are so conveniently spread out. Localizing individual fluorophores is a great start, but how do we localize fluorophores when they're practically on top of each other in diffraction-limited space? That was a real problem to overcome with super-resolution microscopy. So the first person I want to talk about who led us there is Eric Betzig. In 1995, he published a theoretical paper on the idea of using population-based localization methodologies. He suggested that if you have a group of fluorophores, like we see here in B, and you could turn the fluorophores on and off individually, then you could reduce all the fluorophores to single spots over time. The trouble is you would need to have probes that you can turn on and off, and at the time he published this paper, there were no such discovered probes. The principle of his idea was strong, though. In the case of wide-field fluorescence, all fluorescent molecules emit simultaneously, preventing individual molecules from being identified. But if individual molecules could be excited that were sufficiently distant from other excited molecules, they could be fit with a Gaussian function and individually localized. William Moerner provided the next piece of the puzzle when he worked out how to make proteins blink or switch, and in 1997 he published a blinking GFP. So the idea was that we could suddenly do exactly what Betzig proposed. So Betzig came back into the fold and using the photoactivatable GFP showed what we call PALM, photoactivated localization microscopy. This discovery led to William Moerner, Eric Betzig and Stefan Hell receiving the Nobel Prize in 2014 for introducing the world to super-resolution microscopy. So Betzig was able to create this beautiful super-resolution image using PALM with the photoactivatable GFP. But how exactly does PALM work? Well, the key feature of photoactivatable fluorescent proteins is that they exhibit no fluorescence until stimulated by certain wavelengths of light. So initially all photoactivatable fluorescent proteins in the specimen are inactive. I'll use this 3D stack in the middle here to explain the method. Importantly, it goes down on a time axis, so the first frame is at the top where there is no fluorescence. So firstly, a laser of appropriate wavelength from the fluorophore is used to photoactivate a sparse subset of photoactivatable fluorescent proteins, with the number of fluorophores activated being controlled by using a sufficiently weak laser intensity. The fluorescent proteins are activated stochastically, but the probability of activation is proportional to the intensity of the activation laser. Following activation, a readout laser of a longer wavelength is used to excite the photoactivated fluorescent proteins and detect their positions. The subset of fluorescent proteins activated by the activation laser is low enough that the proteins are far enough apart to be individually localized. This frame of data is saved and the fluorescent proteins are left to photobleach until no photoactivated fluorescent proteins remain. The activation laser is then applied again to activate another set of fluorescent proteins, and the method is repeated. So this method of activate, detect, localize, bleach continues until all molecules in the specimen have been localized and none remain to be activated. It's pretty slow, and when they did this work back in 2006 it took 2 to 12 hours to acquire a single image. Since then there have been large improvements in the quality of fluorescent molecules and other advances to speed the process up. Xiaowei Zhuang and her team at Harvard took a different approach to super-resolution because she noticed that some dyes, like Psi5, blink when subjected to a powerful enough laser. So she thought, why not just measure those? There's no need to bleach them because they turn on and off themselves. So she did exactly that and looked at these fluorophores and tried to figure out how to make them blink best. The idea was that if the number of fluorophores turned on is sparse enough to localize over enough frames, all of the fluorophores would have turned on at least once. By eliminating the bleaching step, STORM achieves super-resolution data faster than PALM. So this table shows the localization accuracy of different fluorescence and super-resolution techniques and how they compare to each other. Here on the left is your standard confocal point spread function, and it's roughly, depending on wavelength, 180 to 250 nanometers in XY and 500 to 700 nanometers in Z. Next to that is the STED approach invented by Stefan Hell, who shared the Nobel Prize with Betzig and Moerner. That gives you a tremendous improvement in XY resolution, down to 60 nanometers, but it doesn't do anything in Z. The technique next to it is gated STED, which improves Z somewhat, but what we have now is 3x stead, which would sit somewhere here between storm and sim, and gives you a big improvement in x, y, and z. Sim is wavelength dependent and gives exactly twice the resolution of the best objective you have. It's an optical trick, a very clever optical trick, that allows you to double resolution in x, y, and z. So instead of 180 to 250 nanometers, we're at 100 to 130 nanometers in x, y, and 250 to 340 nanometers in z. Finally, we have Palm and Storm, which have the highest XY resolution of all the super resolution techniques, as well as respectable Z resolution. So what do we need to perform super resolution localization microscopy? Surprisingly, not that much. A modern microscope with focus drift correction is necessary. Localization microscopy involves taking a lot of frames of data and localizing fluorophores to sub 10 nanometer accuracy. You can't afford to drift even a single nanometer away from the starting frame during data acquisition. A laser light source is also necessary. High intensity light is needed to activate some of the photophysical properties of the fluorophores. The fluorophore itself. Fluorophore selection depends on the localization technique used, but there are many factors to take into consideration when choosing a fluorophore, which I will cover in a minute. A sensitive camera. Localization accuracy is directly proportional to the number of photons detected, so the aim is to collect as many as possible. And finally, the reconstruction algorithm. After we've collected all the frames of data we need, we can't possibly manually reduce all those diffraction-limited spots to localized points, so an algorithm does that for us. And that's really it. Super resolution localization microscopy isn't as optically demanding as SIM or STED, for example. Instead, it's more biologically and chemically demanding, especially when it comes to getting the fluorophores to behave. So let's have a look at the fluorophores first. Fluorescent proteins that are used for palm super resolution imaging can be divided into three classes. Irreversible photoactivatable fluorescent proteins, whose fluorescence can be turned on with light of a specific wavelength. Photoshiftable fluorescent proteins, whose fluorescence excitation and emission spectra shift following illumination. And reversible fluorescent proteins, or photoswitching fluorescent proteins, whose emission can be reversibly switched on and off. If we take a closer look at a couple of them, the photoshiftable fluorescent protein monomeric EOS is activated by exposure to UV or blue light, which causes an irreversible spectral shift from a green state to an orange state. It's then excited at 569 nanometers to fluoresce at 581. The fluorophore is then left to bleach. As another example, the reversible photoswitching fluorescent protein DROMPA is activated by exposure to UV light, which makes it fluoresce green. It can then be quenched by prolonged or intense irradiation with green light to return it to a non-fluorescent form. It's then possible to photoactivate it back to the green emitting form with UV light again. DROMPA can actually undergo over 100 cycles of activation quenching with only a 25% loss to the original fluorescence. To explain how storm fluorophores work, it's necessary to use a Jablonski diagram. So typically a fluorophore will be excited and jump from the SO ground state to the S1 excited state, where it will then lose some energy and emit at a lower energy wavelength of light. This is what gives us our Stokes shift from excitation to emission. What that fluorophore can then do is go to this T1 triplet state, the dark state. Here it looks for oxygen-free radicals to bind with, which take the fluorophore back down to the S0 state, and it'll be bleached. But in an oxygen-starved environment, like standard storm buffers, the fluorophore will be trapped in the dark state and can be periodically activated again to emit light, essentially blinking. So as long as there is no oxygen around to blink it, it can keep on going. The most important characteristics of a storm fluorophore are the photon number and the duty cycle. The photon number represents how many photons are put out by the fluorophore, and the duty cycle represents the frequency of blinking. An increased photon output directly correlates to the localization accuracy, and a good duty cycle strikes the balance between the fluorophore being on for too long or too short. Both are equally important when judging the quality of a storm dye, and Alexa 647 is considered to be the gold standard. To highlight the importance of photon number, we can compare Alexa 647 to Atto 655. They have a comparable duty cycle, but Atto 655 has almost a tenth of the photon number. The resulting image makes it clear that Alexa 647 has been localized with higher accuracy, which gives a higher quality image. Now to highlight the importance of the duty cycle, we'll compare Alexa 647 to Psi 5.5. They have high photon numbers, but the duty cycle of Psi 5.5 is almost 6 times that of Alexa 647. The image shows far more spacing between the Psi 5.5 molecules, where less molecules were localized over the same amount of frames. But the most important thing to take away from this is that STORM is highly dependent on the physical properties of the dyes to get high quality super resolution data. The other super resolution localization microscopy technique that I want to mention is DNA paint, which takes the principle of STORM but applies it differently. DNA paint is a newer technique than PALM and STORM, published in 2014 by Ralf Jungmann, and it takes advantage of the complementary nature of DNA strands to produce an effect similar to single molecule blinking. DNA exists as a double helix of 2 complementary DNA strands running in opposite directions to one another. The 2 DNA strands bind highly specifically to each other, so if 2 complementary strands are together in solution, they will spontaneously bind. In DNA paint, one DNA strand of a complementary pair is bound to the molecule of interest, the docking strand, and the other DNA strand is fluorescently labelled, the imager strand. When combined in solution, the imager strand binds the docking strand, producing fluorescent emission of a single molecule at the focal plane. The binding strength and concentration of the imager strand can be tuned to cause repetitive transient binding at a rate similar to blinking dyes seen in STORM. And then, like in STORM, the stochastic binding of imager strands to docking strands on the structure of interest over thousands of frames is reconstructed to create the final super-resolution image. One of the distinct advantages of DNA paint is that it's far less reliant on the physical properties of the dye molecule than PALM and STORM. This allows for multiple dyes to be used for imaging, even those that were not previously applicable to localization-based super-resolution microscopy, allowing the researcher to prioritize photon number when choosing a dye. Multiplexing is also relatively simple. In exchange paint, imager strands in solution interact with their complementary target sequence P1. After the first acquisition round, the P1 imager strands are washed away and P2 images are introduced to image the next target. The P2 images are attached to the exact same fluorophore, but because they have a different DNA sequence, their target is different. This can then be repeated for more cycles, with pseudo-colours being assigned for each respective imaging round. Finally, all rounds are aligned and overlaid to form the final multiplexed exchange paint image of the targets. The image here shows that DNA paint can produce super-resolution images on par with PALM and STORM in multiple colours. But it isn't without its own drawbacks. Unbound imager strands floating around in solution cause elevated background fluorescence, which prevents wide-field illumination of the sample. Instead, you have to do it with turf or light sheet fluorescence microscopy to avoid exciting fluorophores away from the focal plane. It also may not be applicable to live cell imaging because of the unknown consequences of introducing DNA strands into a living cell. Aside from that, it's an excellent localization-based super-resolution imaging technique. So resolution in super-resolution localization microscopy is determined by 2 factors: localization accuracy and molecular density. Localization accuracy is the position of a fluorophore in the lateral dimensions. So, how small can you make a single point? Whereas molecular density is the density of labelled molecules in the sample. Both are equally important when determining resolution. Localization accuracy is determined by this equation, where sigma is localization accuracy, S is the standard deviation of the Gaussian function, N is the number of photons captured, A is the pixel size of the detector, and B is the standard deviation of the background fluorescence. The first term on the right-hand side of the equation represents photon shot noise, the second term represents pixelation noise, and the third term represents background noise from the sample. But with small effective pixel sizes and low background noise, the equation is often simplified to S over the square root of N. To optimize localization accuracy, the most important consideration is to increase the number of photons detected. This is clear from the equation, where increasing N reduces the impact of the first 2 terms on the right-hand side of the equation. This can be achieved with good fluorophore selection and a sensitive camera. Pixel size will have a small effect on localization accuracy, where a larger pixel will introduce more uncertainty as to where the photon landed on the pixel. However, a pixel size greater than the diffraction limit, so an effective pixel greater than 250 nanometers, will become the limiting factor for localization accuracy. An ideal pixel size should ensure correct Nyquist sampling. Background fluorescence is noise from all other sources, including camera noise, scattered light, non-specific labeling, autofluorescence, and other sources of noise produced by the sample. This is typically corrected by diligent sample preparation and choosing a camera with low noise characteristics. Localizing fluorophores to sub-diffraction emitted spots on a pixel can be a difficult concept to follow, as the size of the pixel is conventionally considered to be the smallest area that can contain an image. For example, if we consider that the pixel size has been magnified to approximately 100 nanometers to reach Nyquist sampling, a fluorophore can be localized to a sub-pixel resolve location of under 10 nanometers, allowing multiple fluorophores to be visualized on a single pixel. That seems strange at first, but I'll explain how it's possible. The diffraction-limited spot of the fluorophore extends approximately 250 nanometers in diameter over the fluorophore and covers all pixels in a 3x3 area. The Gaussian fit works by judging the intensity of the neighbouring pixels to localize the fluorophore onto the central pixel. So we can see that the brightest pixel is the central pixel, so that must be where the fluorophore is. The pixel to the left of the central pixel has a higher intensity than the pixel to the right of the central pixel, so we know the fluorophore must be on the left-hand side somewhere. The pixel above the central pixel has a higher intensity than the pixel below the central pixel, so we also know that the peak must be at the top of the central pixel somewhere. By then taking in the information from the corner pixels, the Gaussian fit can accurately localize the fluorophore to a subpixel location. The brighter the fluorophore and the more photons collected, the better the localization accuracy. Of course, a high localization accuracy means nothing if there aren't enough fluorophores present to resolve a structure. According to the Nyquist-Shannon theorem, the distance between two neighbouring molecules should be twice as fine as the desired resolution. This means that if the localization accuracy is 20 nanometers, there should be a fluorophore every 10 nanometers to correctly sample. This relationship between localization accuracy and molecular density is necessary to understand how resolution should be determined in super-resolution microscopy. It's clear from the image that three simulated structures with the same localization accuracy can have a difference in resolution caused by the molecular density. The structure is clearly defined in the image on the right, but not so clear or completely undefined in the images in the middle and on the left. In order to maximize molecular density, the researcher needs to consider how many frames of data are necessary for good super-resolution imaging. This is a video of storm data and represents 1500 frames playing at 45 frames per second. 1500 frames is about a tenth of the amount of frames you would need to even start having good enough frames for good quality data. Mostly you'll be looking at closer to 30,000 frames. So using a standard exposure time of 30 milliseconds, 30,000 frames would take 15 minutes to acquire. The result of the full 30,000 frames is the image on the right, and you can see that almost 3 million individual molecules were localized to produce it. It goes without saying that localization-based super-resolution data is very large in size and takes a considerable amount of time to collect. As we saw in the previous slides, photon number is the most important consideration for improving localization accuracy, which is why a sensitive camera is necessary for localization-based super-resolution techniques. Aside from sensitivity, a faster camera will increase the rate at which you're able to acquire data, and a large field of view will increase the throughput, reducing the amount of acquisitions you need to take. Localization-based super-resolution has mostly been achieved with EMCCDs, which feature high quantum efficiencies and effectively no read noise, so they're very sensitive. Some groups have moved more towards CMOS cameras, which, while not as sensitive as state-of-the-art EMCCDs, have far larger fields of view with much faster acquisition times. However, with the release of the first back-illuminated CMOS device, the Prime 95B, with comparable sensitivity to EMCCDs and superior signal-to-noise, combined with a large field of view and fast acquisition speeds of SCMOS, we found it to give superior super-resolution data when compared to both of these other camera types in terms of localization accuracy, data throughput, and speed of acquisition. So the last thing we need to look at is the reconstruction algorithm, the way we put all of these frames together. Even if we've maximized photon collection and labeling density through good fluorophore and equipment choice, it's the job of the reconstruction algorithm to localize all the points and produce the final image. There are over 50 different reconstruction algorithms out there because there usually isn't one perfect algorithm that best fits every fluorophore, every sample, and every equipment setup. A common one for palm and storm is thunderstorm, which I'll talk more about in a minute, but there are also faster algorithms such as quick palm and rapid storm. DNA paint was developed alongside its own algorithm, Picasso, which also contains DNA paint specific functions such as the ability to design DNA origami structures and simulate DNA paint data. But all algorithms for localization-based super resolution go through a similar set of 7 steps. So firstly, it determines the noise level per pixel. Then it smooths the background fluorescence. Next, it finds the approximate positions of individual molecules. Then comes the subpixel localization of molecules. So this is the basis of achieving super resolution data. This is where it localizes every point spread function of every fluorophore and every frame of data. It then has to overcome the crowded field problem, where multiple fluorophores exist in the same spatial region and need to be individually localized. Post-processing allows the user to correct their data by doing things like removing molecules with poor localization, merging reappearing molecules, removing duplicates, and drift correction. And finally, it all ends with a visualization of the final image. So following on from this, once the final image has been produced, localization microscopy is an interesting one to analyze, because in many cases what you're seeing isn't actually a real image, not in the conventional sense anyway. One of the reasons localization microscopy images look so nice is because our eyes do extra reconstruction work for us. The eyes and the brain are particularly good at recognizing patterns, so when we see an image of something like a microtubule or other filamentous structure, we can't help but fill in the gaps. But when the image is zoomed in, we can see that there are actually quite a lot of gaps. It's reminiscent of a Seurat painting, where he hasn't actually painted a real image, but our brain is filling in the gaps for us. This is something that researchers need to think about when deciding if super resolution is necessary for their work. To conclude this talk, a question that should be asked of all techniques is what they've enabled us to achieve that wasn't possible before, and super resolution localization microscopy has provided us with a wealth of new information. For papers concerning the usefulness of super resolution localization microscopy and the development of the techniques, we recommend the following papers that investigate the molecular organization of focal adhesion complexes using PALM, the distribution of integrins inside focal adhesions also using PALM, the molecular organization of the Y complex within the nuclear pore using STORM, arrangements of subunits inside skeletal septin filaments in cells also using STORM, and finally, how super resolution localization microscopy techniques have provided insights into HIV biology. This is an excellent review with both PALM and STORM included. Thank you very much for listening to this presentation. If you want to learn more about super resolution localization microscopy as well as find information about choosing the right camera, please follow links on this page for relevant application notes and technical notes. You can also navigate to other sections of the website where you'll find similar information for other applications and techniques. Thank you very much.